He's going to be drilling us about transporters and how they really work. And there's a lot of depth to transporters, and Rob knows it all. Everything from the vesicular, the membrane.
So I'm going to talk. Thank you, Chris. Thanks for the invitation. The first part is just going to be, as I've done before, maybe I'll have to change this in the future, but is about just how transporters work, and actually some of the transporters that probably people interested in addiction care the most about are among the most poorly understood. So, although there's lots of interesting things. So I'm going to just talk about transporters for the first part, and the second part will be about glutamate release from basically midbrain neurons, in particular dopamine neurons and other neurons. Okay, so that may be a little more closely aligned, but I thought this has been of interest before. So, and a lot of people in neuroscience know about channels. Transporters are completely different, basically. People think that channels basically move ions very quickly. Transporters are kind of slow. Channels do not saturate with high concentrations of ions. Transporters are like little enzymes that move things across the membrane, so they saturate often at low concentrations of the substrate. Many neurotransmitter transporters are pretty high affinity. And of course, channels really can't concentrate an ion, but channels can't concentrate an ion, but transporters can certainly concentrate their substrates. And the basic difference between channels and transporters is that channels are a pore that is continuously open from one side of the membrane to the other, and transporters do this alternating access, where they're open to one side, then open to the other, allowing the substrate to get across the membrane. And this very simple difference, alternating access versus a continuous pore, predicts almost all of their properties. So I'm going to illustrate some of these things and try to bring up the points that are relevant for the transporters, for the neurotransmitter transporters. So alternating access. Okay. So this is basically it. And so basically what happens in a very simple transporter is the substrate hops on, is moved across the membrane, comes off on the other side, and then to get net flux of a substrate, you actually have to move the unloaded transporter back to the first side of the membrane. This is very important. Actually, this is even important for how amphetamines work, as I will mention in a minute. So to get net movement of the substrate, you have to have movement of the loaded transporter and the unloaded transporter back in the opposite direction. And there are examples, and many transporters will do this, and some transporters only do this, where they only move the substrate, and they never move, the unloaded carrier never reorients across the membrane. So basically all they do is exchange substrate on one side of the membrane for substrate on the other side of the membrane. No net flux. And this is basically one of the ways in which amphetamines are thought to work. So we'll talk about that more in a minute. But just to illustrate some of the weird things that happen. So this is an example of what I just showed you. This is an experiment where you take vesicles expressing a glucose transporter and you load them up with radioactive glucose. And then you dilute them into medium without any glucose, and you see how fast the radioactive glucose comes out. And the question is, if you put unradioactive, non-radioactive glucose on the outside, does it slow down, do nothing, or stimulate the efflux of the radioactive stuff? Most people will say it will slow it down. You're putting stuff on the outside, it's just going to come out more slowly. In fact, what happens with transporters is putting the substrate on the other side of the membrane over here accelerates the efflux. The reason for this is the exchange reaction. So that's what I was just showing you. Basically what's happening is if you've got radioactive stuff, let's say the blue substrate on the inside, and unradioactive stuff on the outside, what happens is the radioactive stuff goes out, the non-radioactive goes in, and all you're doing is exchanging the radioactive for the unradioactive, and this is often much faster than going around the whole cycle. So actually, these exchange reactions are faster but there's no net flux. And basically, this is what amphetamines are thought to do. This is what they were classically thought to do, is that amphetamines on the outside of cells, on the outside of monoamine neurons, would go into the cell through, for example, the dopamine transporter, and the dopamine on the inside would come back and go out, and the amphetamine, and they would keep exchanging. And the advantage for amphetamines was that amphetamines are lipophilic, and they would, every time they would go in, they would diffuse across the membrane back out and keep doing the cycle, which would result in the massive efflux of dopamine. So this is one of the older models of how amphetamines work and why they're so powerful, in fact. So,
Is that still pretty?
no, there's newer ideas about how it works, but this is, and there's probably a lot of truth to that too. But I will talk about that later if I don't. I have some slides on that. So this is exchange. Some transporters only do exchange normally. That's all they do, exchange one thing for another, no net flux. But many transporters will do it under certain circumstances, and some drugs cause them to do it, like amphetamines. Okay. So just a little overview, more overview of transporters. You know, there are pumps which make basically ionic gradients in most cases, which do a lot of important things in the cell. We're not talking about a light-driven pump, but you know, the ATP-driven pumps make the ionic gradients for sodium, potassium, chloride, everything in everything that cells do across membranes, basically. And then those are pumps, and then there are transporters which couple the movement of ions made by these pumps. The gradients are made by these pumps, couple the movement, the downhill movement of those ions to the movement of substrates off against their concentration gradient. Okay. So, okay, this is the sodium-potassium ATPase that makes the sodium and potassium gradients. They're called P-type ATPases because actually a step in the reaction cycle, the transporter actually gets phosphorylated. So it's called P-type ATPases. The structures of these have been determined just within the last few years, so we actually have learned a lot about how they work. And the other one, which is of great interest to what we work on, which is the transport of neurotransmitters into vesicles, depends on a proton pump. So they're actually two closely related proteins. One you probably all know about, which is what makes ATP in cells. So this is the mitochondrial ATP synthase, which the electron transport chain, as remember, is pumping protons out of the mitochondria. This lets the protons run back into the mitochondria, and this thing spins around and produces ATP. It's a little rotary machine. You can put actin filaments on them and see them spin in a microscope. And very closely related in sequence, but entirely distinct, it's a completely different set of proteins, is involved in pumping protons into synaptic vesicles, dense core vesicles, and lots of other endosomal membranes in cells. And it looks exactly like this, but it works exactly the opposite. What it does, this is the vacuolar type proton ATPase, it uses the ATP, the energy stored in ATP and ATP hydrolysis, to drive the movement of protons into the lumen of these vesicles. So you can see here, protons are driving the production of ATP in mitochondria. Here, ATP is driving the movement of protons into vesicles. Very similar, but distinct. Okay. And so getting a little bit closer to the neurotransmitter transporters, basically all of the ones at the plasma membrane rely on a sodium gradient. Some are a lot more complicated, but this is the bare bones version of them. So basically what they do is this sodium gradient. Sodium runs downhill into the cell, down its concentration gradient, and to some extent its membrane potential, and coupled to it is the movement of substrate. And the way these transporters work is the transporter will not move things across the membrane unless the sodium is there and the substrate or the neurotransmitter is there. They both have to be present, then it moves to the inside. So it's like a little logic gate where you have to have both things present, then they go across. And you can see why, you have to have both present. If it would go across with just the sodium, that wouldn't be very good. You'd lose all your sodium gradient. Okay. So this is just to see if this makes sense to you. So if you've got a transporter like this that's moving substrate from the outside to the inside, coupled to the movement of sodium, one substrate, and you have, let's say, a 10-fold gradient, more sodium outside than inside by 10-fold, how much substrate do you get on the inside at equilibrium? You get 10 times more outside or inside? Inside. Thank you. Yes. So it's actually this is the simpler one. So basically the
You can answer the difficult.
yeah, the more difficult one, which is the way that most of the proteins we're interested in work. But the simple one is that at equilibrium, the forward reaction is equal to the reverse reaction. The forward reaction is dependent on the concentrations of the substrates on the outside, and the back reaction is dependent on the concentrations of the substrates on the inside. So if you set those equal, so the forward reaction equals the reverse reaction, and then you rearrange it, the sodium out over in, which would be tenfold, let's say roughly, will give you a substrate concentration gradient of tenfold in over out. This is exactly what Chris said. So it's just from these equations. Okay. And an exchanger will do something different. So this is more like what neurotransmitter transporters do. So here you can vary the stoichiometry of the sodium. Most of the transporters we're interested in move more than one sodium. So if there's two sodiums, okay, this is the difficult question. If there's two sodiums, and let's say you have a tenfold gradient of sodium outside over inside, how much concentration of the substrate do you get?
I don't know. Hundreds.
Thank you. Chris, you are on a roll. It's basically the same thing as what I said. I have the equations here. So actually, basically the forward reaction is now the square of the sodium concentration, and the reverse reaction is the square. And you just rearrange these. And now, if you have a tenfold gradient of sodium, so it's log to the 10 of, so this is 1, log to the 10 of 10 is 1, times 2, because you have two sodiums being moved, you get 2 log units of substrate. So 10-fold of sodium will now give you 100-fold of substrate. If you have 3 sodiums, how much concentration of the substrate do you get? 1,000-fold. Okay, good. Okay. So
That was easy.
okay. So yeah. So now this gets a little bit more complicated. I realized when I was doing this I probably should stay away from this, but most of these transporters that we study are also dependent on the membrane potential because they move net charge. So what does that do to how much concentration of substrate you get? Remember, the inside of the cell is pretty negative, like minus 60 millivolts, just say for simplicity. How much will that concentrate the substrate? The bottom line is to figure out what actually happens to the substrate. It's the, this is what I showed you before, the sodium gradient, and this is the effective membrane potential. And basically, ZT is the number of charges moved. Let's say it's 1. Let's say you have 2 sodiums and 1, like say glutamate. So 2 sodiums, 1 glutamate, it's 1 net charge going in, 1 plus. And if the cell is minus 60 millivolts, what that's going to do is it's going to concentrate the glutamate tenfold. That's basically what this says. You just add this on. That's an additional log unit of substrate. So this would be 100-fold if there's 2 sodiums, and this would be another 10-fold because you're moving 1 charge. So this would be now 1,000-fold. The effects of the concentration gradient of sodium, the effects of the membrane potential. And, okay, I don't want to belabor this. Okay, what does this mean? And we'll get into some specific examples. And actually, for the transporters that, again, we care most about, a lot of this is not well understood. So I'm going to focus on the ones where it is well understood, other neurotransmitter transporters. But briefly, so, the idea is basically, as you know, that the plasma membrane neurotransmitter transporters are there to clear the transmitter after it is released by excitatosis. Okay, so that's pretty straightforward. And old experiments done, you could do experiments with cocaine, but some of these very nice experiments done by Mark Carone many years ago now. This is in striatal slices. You stimulate them. You measure dopamine overflow with a carbon fiber electrode, and you stimulate a burst, and this is how much dopamine you get. And if you don't do anything, the dopamine is cleared very quickly. If you look in the DAT heterozygotes or the DAT knockout mice, now you stimulate, you get the dopamine released, and it is cleared. This is diffusion, basically. So as people had predicted, dopamine is cleared by the dopamine transporter, and if you don't have it, it's basically cleared by diffusion in general. Now, the other thing that they noticed, which was very unusual, is in the DAT knockout, they measured the amount of dopamine in the brains, in the striatum. It's about 5% of normal. So the other thing that these transporters do is they recycle the transmitter so that it can be released again. Obviously, in most cases, the transporter is expressed by the dopamine neurons, so it can take up the dopamine, put it back into the cell, and put it back into vesicles so you can release it again. And this is
that why the peak is lower in the
That's probably why the peak is, the levels are in the stratum are 5%. It's amazing there's this much. It's amazing it's as good as this. But yeah, so it has a huge effect. In other places in the brain, the transporters have a little bit less effect. So in the cortex, where the dopamine projection is a little bit less dense than on the striatum, diffusion probably plays a larger role. But in the striatum, where the terminals are very dense, the transport is probably very important. It's particularly important for clearance. Okay. Okay.
So how many shots would a neuron have with the transporter?
Well, you know, if you look in striatal slices, what people have done, they repeatedly stimulate, and they say, actually, if you put the pulses more than a minute apart, you see depression. You actually have to space them a minute apart to start actually having maintained their response. But if you do it in vivo, I mean, we haven't done these experiments, but if you do it in vivo, you can do it much more frequently. So it's not totally surprising. There's sheared off terminals in slices. But, you know, people look at sheared off terminals in hippocampal slices too. And you can stimulate them a lot of times and they keep on responding. But there's something different about these. And the striatal slices just doesn't seem to work that well. But anyway. Okay. So this is the glycine transporter. And again, maybe, you know, I don't know how much of this I really should go through. But I thought I would give you some examples of transporters that are well understood. And one of the best ones is the glycine transporter. So these are just examples of experiments people have done. There are two glycine transporters. One of them is expressed in glia, that's GlyT1, and there's another one that's expressed in glycinergic neurons, which is GlyT2. And in this paper, this is work of Stéphane Souplisson from a few years ago now, looking at the properties of GlyT1 and GlyT2. And you can take these transporters, express them in xenopus and add glycine and see inward currents, because when the glycine is going into the cell, there's net positive charge being moved in, and you can measure these currents. At the same time, they can use radioactive glycine and actually figure out how many picomoles of glycine are going into the oocyte, measure the radioactivity. So what is the purpose? I should just back up for a second. What's the purpose of these experiments? To determine the stoichiometry of transporters, because their stoichiometry determines how much of a concentration gradient they make of the substrate. And so if they have a really great big stoichiometry, they're going to make a huge gradient. And you might think, I'm going to try to illustrate here why this is particularly important. So these two transporters, as it turns out, differ in their stoichiometry. So GlyT1, again, you add glycine, currents go in, the substrate glycine goes in. GlyT2 looks very similar. But if you look at the curves, the picomoles of glycine that go in and the picomoles of charge that go in, basically, the picomoles for GlyT1, which is what's shown over here, the picomoles of charge are equal to the picomoles of glycine. So there's one glycine for one charge going in. The bottom line is GlyT1, there's two sodiums going in. For GlyT2, which is the one over here, there's 2 picomoles of charge for every glycine that goes in, because there are 3 sodiums going in with the glycine. So that's all the differences between these two. 2 sodiums versus 3 sodiums. Okay. And I think I'm going to abbreviate this, because it is. And I'll just get to this. Okay. So what is the gradient predicted for GlyT1? So GlyT1 is the one that's actually expressed by astrocytes. It does 2 sodiums, 1 glycine, and 1 chloride, and moves 1 net charge because of that. GlyT2 is the one on glycine neurons. It's 3 sodiums, 1 glycine, and 1 chloride. It moves 2 positive charges along with 3 sodiums. This one, the one on neurons, is predicted to make a gradient of 1,000,000-fold outside in the cell compared to outside. And the astrocytic one makes a gradient of a mere 10,000-fold. So what's the difference between these? So several years later, actually Heinrich Betz at Frankfurt, he knocked out GlyT1 and GlyT2, and they have completely opposite phenotypes. Okay? So GlyT1, if you knock it out, the astrocytic one, what happens is it's the brain, the animals behave as if they're in glycine coma. They have vast excess of glycine in the extracellular space, and it's as if their glycine receptors are being massively stimulated. GlyT2 knockouts, the ones over here, they behave as if they have no glycine receptors, as if they don't have any glycine, because they can't put the glycine into the nerve. They can't take it back up, so that the glycine can be put into vesicles. So there is no neuronal release of glycine from the GlyT2 knockouts. So 2 different transporters, and expressed by different cells, different stoichiometries, and completely different effects on physiology.
Ron,
Yes.
what is the consequence on behavior?
Well, I mean, the GlyT2 are the ones that are basically resemble glycine deficiency. So they have a startle. So these animals that don't have glycine receptors, if you startle them, they just fall over. They drop, they keel over. There's a certain spontaneous mutants in livestock that have this. So that's how they behave. The others are basically in coma. GlyT1. So a completely different thing. Okay. So just to get back to the transporters we care about. So DAT, the dopamine transporter, again, it's a member of the same family as the glycine transporter. It's very closely related in sequence. So is SERT and many other neurotransmitter transporters. It moves either 1 or 2 sodiums. I'm not entirely clear from the literature. Dopamine, which is positively charged, and 1 chloride. And that's what it does. And the serotonin transporter, which is really closely related to the dopamine transporter, does something a bit weird. It moves 1 sodium, and it moves a potassium in the opposite direction. So this one is moving, let's say it's moving 2 sodiums, 1 chloride, 1 dopamine. It's moving a bunch of positive charge into the cell. And yeah, nice. And then this one, SERT, it's 1 sodium, 1 chloride, 1 dopamine in exchange for potassium going out. This one moves no charge. I have to say, I don't work on these particular transporters, but the bottom line is that this one is electroneutral. It doesn't respond to membrane potential at all. I have never understood why the serotonin transporter is sort of wired up differently from the dopamine transporter, but I think it's a really interesting question for people, working in this area.
So do we know whether, in reality, this is a series of conformational changes that will either happen in series or nothing happen? Why do we need these 5 steps? It's because the conformational changes are all correlated with each other?
Basically, the way I look at it is the ions are binding and they're putting energy into the system. So when you have 1, 2, the more sodiums you have binding, the way you can think of it intuitively, is they're like ratcheting on the crank to put, tighten on the spring or something to put power into the system. And then when the substrate comes, it can be released. So that's the way I think of it. Obviously, you could make a huge gradient with 10 sodiums, but it would take forever. The whole thing would slow down. It wouldn't be very efficient. But it's putting energy in to make a big gradient. That's
Say if
basically
it's co-releasing an amino acid like glutamate, would that impinge or could that influence the
I'm going to talk about all this kind of stuff. So yeah.
So Robert,
Yeah.
maybe what Anto's also
Yeah.
asking is, do we actually know physically the physical basis of some of these intermediates?
The structures of some of these have been determined. So these guys all belong to one family. Eric Guo solved the structure. It's called LUT from bacteria, and a lot has been learned about that. And there's a whole other family, which I'm going to mention briefly now, the glutamate transporters, and their structure also solved by Eric Guo. So we do know a fair amount about it. And this is actually, thank you for asking, this is the other family of transporters. So everything I've talked, all of the neurotransmitters are carried by the one family I've just talked to you about, DAT, SERT, the glycine transporters, norepinephrine transporter. And there's a separate family dedicated to glutamate. Okay. So these are the glutamate transporters, and they have a quite amazing stoichiometry. They move 3 sodiums, 1 proton, and glutamate in exchange for potassium going out. They make gradients of probably over 1 million-fold. So they are particularly powerful. And most of these are expressed by astrocytes. A few are expressed postsynaptically. Very few, if any of these, are expressed on glutamate terminals themselves. It's a bit of a mystery. So the glutamate, when it's released, is not taken back up into the cell that's released it. It's taken into astrocytes and shuttled back into neurons through more complicated mechanisms, which I'm not going to talk about. Okay, and here's some other interesting properties which are relevant, and I'm going to talk about how they're relevant for dopamine transporters in a minute, but I wanted to talk about the better understood case first. So this is glutamate transporters, and I don't know how many of you have a background in physiology, but basically these would be, for example, oocytes expressing EAT1, excitatory amino acid transporter 1, or EAT2 or EAT3. And then the bar is where glutamate is added to them, and these are the currents. And actually, just look at EAT2 here. This is the typical one. You add glutamate to the outside of this oocyte, and you get inward currents because charge is moving into the oocyte. Fine. But this is very weird. EAT1, you add glutamate to the outside, and now currents are moving in both directions. Okay? If it's a transporter, this is a transporter. if you add the substrate on one side, it can only move the transporter in one direction. How is it that adding the substrate on the outside of oocytes expressing this transporter get the current to go in both directions? This is crazy, okay? That's more like a channel, and that's actually what this is. So basically, the glutamate transporters we know now, 3 sodiums, glutamate, protons, potassium, and in one stage of the transport cycle, it's associated with an opening of a pore, which actually lets chloride through. These transporters don't move chloride as part of the transport cycle, but they move chloride as part of a channel, which is associated with their activity. Okay, and this is very clear for these transporters. It's very well worked out, and in fact, in some places, this glutamate transporter behaves as a receptor for glutamate. So glutamate binds to it, opens a channel in the retina of some amphibians. These transporters actually serve the purpose of receptors. So this is a protein which does 2 different things, channel and transporter. And this is an example of what actually happens. So now we're going to get a little bit into the dopamine transporter. So it's actually very hard to see currents associated with the monoamine transporters on the plasma membrane. Really hard. So they're not as easy to study using these nice methods. But Aurelio Galli at Vanderbilt has been able to see currents associated with, in particular, the dopamine transporter. So these are HEK cells expressing the dopamine transporter. And what he does, it's a little artificial, but you basically patch onto the cell with a pipette that contains 2 millimolar dopamine. That's a lot of dopamine. Cytoplasm of neurons probably has vanishingly small sub-micromolar amounts, but 2 millimolar in the pipette. And then he depolarizes the cell. And he probably has some sodium in there, which I forgot to mention, basically to try to get the transporter to go the other direction. So instead of taking dopamine up into the cell, now he wants to see, can he release it? So when he depolarizes the cell, these are cells expressing the dopamine transporter, and basically what he sees is outward currents. So he's making the inside positive, the dopamine and the sodium and everything is going out, and he thinks that this is actually, the dopamine is leaving the cell through a channel, which is part of the transporter, but not exactly related to its transport activity, but actually a hole in the transporter that lets the dopamine out. And he actually can measure the dopamine that's coming out of these cells by amperometry. And when he depolarizes the cell, he can see the dopamine coming out here. Okay, so that's some evidence, and he thinks that this is not actually related to transport. And this is where it gets interesting in terms of people interested in drug addiction is they also did some experiments where they just cut off the N-terminus of the dopamine transporter. This is not the part of the protein that's thought to be involved in transport activity. And when they did the same experiment and they expressed it and it was expressed perfectly well, they repeated the experiment, there was very little dopamine coming out here. And what was subsequently found is that the N-terminus of the dopamine transporter can be phosphorylated, actually by CAM kinase, which also associates with the transporter. And it's the phosphorylation of the transporter at the N-terminus which enables it to do efflux. So this has given rise to a very interesting idea, which I'm trying to summarize now. Actually, this is independent of transport. So they can make mutations of the N-terminus of the dopamine transporter. It does not affect the uptake by the transporter of dopamine. They take up dopamine perfectly well. But what those mutants do when you either delete the N-terminus or block the phosphorylation is it affects the ability to do efflux. So they're saying there's an uptake mode and an efflux mode, and they're separable. And the interesting thing about this is, most of you probably thought of these proteins as the targets of drugs of abuse. And that's what drugs work on, and then it unleashes a series of events which eventually results in addiction. But what this is saying is actually that modification of the site of drug action itself influences its behavior and would influence the subsequent ability of dopamine and cocaine and amphetamine to induce the efflux of monoamines. So in other words, there's modification of the site of drug action such that you're going to influence the future effect of the drug. So it's still not totally clear what's going on here. But there seem to be 2 different modes. And so I talked before about exchange, amphetamine exchanges for dopamine to release dopamine from neurons. But this is an additional effect which seems to be required for dopamine efflux in response to amphetamine, is you actually have to phosphorylate the N-terminus of that so that amphetamine can actually do this. If you have these mutants, amphetamine doesn't work very well. So that's the interesting thing. The exact mechanism for this is not understood, but there is clearly the potential for regulation. And again, it seems independent of uptake, which is amazing. Okay, but I'm summarizing. This is not my data, but it's very interesting and it's very provocative, and so.
I would think that the flux that you could get through this mechanism
Yeah.
would be higher than the flux that you would get from
Potentially. The question has always been, how much dopamine do you actually get in the cytoplasm of a dopamine neuron? Most of it's in
Right.
vesicles.
Right.
And could this actually happen? But they did experiments in neurons, and they saw something similar with real dopamine neurons.
But you also affect the vesicular transport of that.
With amphetamines, yes. So that's probably crucial for it to work, is the amphetamines release the dopamine from the cytoplasm, from the vesicle into the cytoplasm, which we'll talk a little bit more about.
So
Okay.
have they got a phosphor-specific antibody for this?
I don't recall. I'm not sure they do. They've looked at a lot of things of the phosphorylation of the N-terminus. It requires phosphorylation at multiple sites, but they may at this point. I'm not sure. Okay.
You know, Charlie brought up things,
Mhm.
you know, with the idea that transporters can be
Mhm.
inserted and removed
Yes,
from
yes,
the membrane,
yes.
so there's dynamic control there. And
So there's been
suggesting
huge
that
amount
there's
of
a lot
interest,
control.
as always, in the trafficking of these proteins, and we study trafficking too. But what I've particularly liked about this is it isn't trafficking. It's the actual behavior function of the protein. So, and there are not that many examples of that. So,
Yeah, it's more like what
Yeah.
people talk about with receptors that can be
yes,
modified
that's
in
right,
various ways.
and to
So
do
that's
different
the
things.
same
Exactly,
kind
exactly.
of
Yeah,
overlay on the transport.
yeah. Okay.
It'd be interesting to know if drugs of abuse,
Mhm.
particularly those that affect transport activities,
Mhm.
have effects on these mechanisms. So, for example, if they produce some sort of persistently phosphorylated
Right. Well, I
state
think
or
that is
something like
that's
that.
sort of the idea, is that the drugs of abuse will activate the kinases which phosphorylate that, which will then modify the transporter so that in the future the drugs have different effects. Yeah, or maybe greater effects. In this case, phosphorylation is actually required for the efflux. So in this case, you would make the transporter more susceptible to releasing the dopamine later on. Okay. And so this is sort of the introduction to what I'm going to talk about. I've talked about the plasma membrane transporters. What we work on is the vesicle transporters, loading neurotransmitter into vesicles. I mean, this is very important to terminate the signaling by the transmitter and to recycle it. But if you don't have it in vesicles, it can't do anything. So this is what we work on. And actually, this is a little bit introduction to the second part now. So, and this gets into the question that Paul asked before. So this is the transport of all classical neurotransmitters into vesicles is driven by this proton pump that I talked about that spins around. It uses ATP hydrolysis to pump protons into the vesicle, and it makes a pH gradient across the vesicle, and it makes the vesicle positively charged. And what's interesting is that different classical transmitters depend on either the pH gradient across the vesicle or the membrane potential. And so monoamines and acetylcholine depend on the pH gradient. Glutamate depends on the membrane potential, and GABA is somewhere in between. And it sounds like a minor difference, pH gradient, membrane potential, who cares? Well, these proteins, these three different sets of proteins, are completely unrelated to each other. So they're catalyzed in completely different ways. These are sequence unrelated to these, which are unrelated to these. And one of the big questions, very simple question, is what then regulates the pH gradient versus the membrane potential of the vesicle? If you make a pH gradient, you're going to help the vesicle to fill with monoamines, not with glutamate. If you make a membrane potential, you're going to help the vesicle to fill with glutamate, not monoamines. So what regulates the expression of this gradient as either a pH gradient or membrane potential? And that's the first thing that I'm going to be talking about for dopamine and glutamate. Actually, probably acetylcholine, serotonin, norepinephrine, and glutamate. Probably everything, all the cationic transmitters. So, any quick questions about this before I move to the second part? We won't take a break because that was quick. Okay. Okay, so okay. Many years ago it was shown that actually serotonin neurons and dopamine neurons, when you put them in culture, that would form glutamate synapses onto themselves. And people said, well, this is just an artifact of culture, and it's not really, this doesn't really happen in the brain. So there were a lot of questions. Steve Rayport at Columbia cut some slices that went through the midbrain into the ventral striatum, and he stimulated the cell bodies of what he thought was the dopamine neurons, and he recorded fast glutamatergic EPSBs in the medium spiny neurons. So that was some evidence in vivo that dopamine neurons could release glutamate. But there were questions about it. The responses were very small, and so people weren't sure. So what was found is that midbrain dopamine neurons do transiently express the vesicular glutamate transporter, VGLUT2. So this is one of these 3 proteins that I mentioned briefly before that pump glutamate into the vesicles. There's 3 of them for glutamate alone. And VGLUT2 is expressed transiently during development in the dopamine neurons. And so the question is, that we were going to ask by manipulating VGLUT2 is, do dopamine neurons really release glutamate in vivo? And what does it do? And we don't have the answer to all of these things yet, but we have the answer to some of them. So what Tom Nasco in my lab did is he made a conditional knockout of VGLUT2. Actually, he started when he was in Richard Palmer's lab. Charlie mentioned him briefly before, because he had done work as a student showing that dopamine could not account for some of the rewarding effect of opiates. So he was interested in whether glutamate might be responsible for some of this. So what he did is he made a conditional knockout of VGLUT2, and then he crossed it to mice expressing CRE in the dopamine neurons, that CRE mice, and to specifically inactivate VGLUT2 in the dopamine neurons. Now, I should add, if you inactivate VGLUT2 globally, the animals die at birth. It's half the brain, the brainstem, okay? It's everything. But he made these mice, and this was a very specific lesion, loss of VGLUT2 in this very specific population of cells with it. And of course, they're supposed to release dopamine, so I thought, oh God, we're not going to find anything. It'll be the opposite. Maybe they don't really do this. But we did find a few things, and first, we did some simple behavior, and we've done more, but it's still sort of in the process. And these things didn't really show any effect of the conditional knockout of VGLUT2 and dopamine neurons. Rotor rod, spontaneous activity. But we did find that when we gave them cocaine, and another group did something similar with amphetamines, that they have less of a response, considerably less locomotor response to cocaine and amphetamines than the wild type, the controls in these experiments. This is sensitization. They start out at lower levels, and they eventually sort of catch up to the sensitization and the controls. So they can do that, but they start out at much lower levels. So, okay, they have less response to cocaine and amphetamines. We wondered, well, why is that? And there are several possibilities, but one simple possibility is that they don't release as much dopamine. Remember, so what cocaine does, of course, is it blocks the reuptake of dopamine. And if there's less dopamine being released through vesicular exocytotic mechanisms, cocaine won't do as much, because it can't block, there's less to block the reuptake of. So we thought that might be a possibility. And so what Tom did is, first in the dorsal striatum, he measured the level of dopamine and its metabolites, and there was no difference between the controls and the knockouts in the dorsal striatum. But in the ventral striatum, there was a substantial, about 30-40% reduction in dopamine levels in some of the metabolites, specifically in the ventral striatum. And this was convenient because the VGLUT2 was shown to be expressed specifically by VTA neurons, not substantia nigra neurons. So this was consistent with an input to the ventral striatum. So, so that's what he found. And the question is, why is this? Why is the dopamine reduced? And this is still an ongoing controversy. Some people have suggested, we have not gone back and checked this ourselves, is that there are fewer dopamine neurons in these mice for some reason. A small reduction, probably not as big as the reduction in dopamine we saw, but maybe there's a small reduction. We measured TH levels by quantitative Westerns, by measuring L-DOPA in vivo, and all sorts of things. No change in the conditional knockout, in the level of tyrosine hydroxylase. When we looked at the anatomy of the projection from the midbrain, the mesostriatal projection, everything looks okay by light microscopy when you stain for tyrosine hydroxylase in the knockout compared to the wild type. But I should say that this is still a major unknown. The expression of VGLUT2 is particularly high in these cells during development, and then it goes down. I'm going to show you experiments we've done in the adult, but its real role in development, we still don't really know. Grossly, there's no obvious defects.
So I missed, how
It's DATCRE, yeah.
Oh, Jeffrey, sorry.
DATCRE, yeah, yeah. So, we didn't see anything with these, but again, I'm not sure that there wasn't some change. But we were left with one other possibility, which is it's been known for a long time that to make the pH gradient that drives the uptake of monoamines like dopamine, basically the proton pump, if you just take synaptic vesicles and add ATP to them, they actually don't make a pH gradient. They will not do it. What has to happen is chloride has to be added. And the reason for this is the proton pump pumps protons in here, and it makes the vesicle very positive. And after just a few ions, just like in any membrane, makes it so positive that the proton pump can't pump any more protons in, and you get no pH gradient, effectively no pH gradient. And the only way you get a pH gradient is chloride goes in to neutralize the protons so the proton pump can keep pumping, and then you get a pH gradient. This kind of thing has been known for a long time, okay, that chloride does this. And what we started to wonder was, does glutamate do the same thing? Okay? And so that basically, in these vesicles, we would add ATP. You'd get a membrane potential. Then instead of chloride, what chloride would do is it would dissipate it, but maybe glutamate would do the same thing, dissipate the membrane potential and enable the proton pump to put more protons in, and that glutamate would do exactly the same thing as the chloride. So this is kind of weird because chloride is thought to go through channels, it's thought to be powerful and do all these important things, and the glutamate transporter is this little clunky thing that's moving glutamate into the vesicles. Go ahead.
Well, does VGLUT2 co-localize the same vesicles?
We did that. I wasn't going to show you that. Yes, it does. At least some of it is there. At least some of it. This is an additional point, but at least some of it is definitely there. We did immunoisolations and stuff, but I have functional data, which I think is the most convincing. So, and the idea is that glutamate does the same thing. So, and we would have been surprised. Why would you have glutamate do this, going through this slow, clunky transporter, as opposed to chloride, which should go through some channel or something and be much faster. So, but we did the experiments, and these were the shocking experiments. This is a way you can measure the acidification, the pH gradient developed in vesicles. It's quenching of a dye called acridine orange. Lower down means the vesicle is becoming more acidic. Protons are going in. And so you can see here, we add ATP to synaptic vesicles, very little acidification. We add a little bit of chloride for technical reasons to activate the V-glutes, technical. And then we add either 4 millimolar aspartate, chloride, and glutamate. And I should add, these transporters are exquisitely specific for glutamate. They don't recognize aspartate at all. So aspartate is the control for glutamate. And what we found is glutamate at 4 millimolar was better at acidifying the vesicles. Certainly aspartate didn't do anything. And chloride didn't do much either. It was better than chloride, than the ion. And this is a dose response. So at high concentrations, chloride will actually be better, but glutamate is better at low concentrations. It saturates, consistent with the Km of the V-glutes. This is all consistent with the behavior of all the V-glutes. They saturate right around here, 1 to 2 millimolar. And this is what we saw. So this was evidence that glutamate was actually very good at acidifying vesicles. These are the functional experiments. So these are synaptic vesicles where we have 2 millimolar chloride here, and we're looking at the uptake of serotonin. It could be dopamine, but serotonin is cheaper and more stable. So this is synaptic vesicles, and this we've loaded them up with, this is uptake in the presence of glutamate and aspartate. Remember, glutamate is recognized by V-glutes, aspartate is not, so that's why it's good control. And there's clearly a little bit more glutamate, monoamine, serotonin uptake when we have glutamate present than we do not have glutamate present. But we use really low amounts of chloride here, because obviously chloride could do this too, glutamate could do this. If we have chloride there, maybe glutamate won't do anything, so we specifically used very little chloride for these experiments, and you can see glutamate has an effect. But then this is what chloride alone does, 2 millimolar chloride, 20 millimolar chloride. Chloride alone can certainly do something at least as good as this, and we thought, well, physiological chloride in cells is about 20 millimolar. So let's repeat this experiment and see if glutamate does anything in the presence of normal amounts of chloride, like 20 millimolar. We thought, oh, now we're not going to see anything. But the amazing thing is that even at high chloride, glutamate has exactly the same effect. It's as if it's almost acting semi-independently of chloride. So this is functional evidence that they're on the same vesicle, because glutamate helps to fill the vesicle with serotonin or dopamine. And I think this is basically looking at acidification again. 4 millimolar glutamate, 20 millimolar chloride. You put them together, you get an additive effect. They're doing something together. And this is the real explanation for it. This is a drug called baflamycin. It stops the proton pump. Can't put any more protons into the vesicle. And now we did this with vesicles that we had acidified with either chloride or glutamate. 14 millimolar chloride, 4 millimolar glutamate get exactly the same pH gradient down here with acidification. Then we inhibit the proton pump, and the vesicles that were acidified in chloride lose their protons really fast, and the vesicles that were acidified in glutamate lose them much slower. So the vesicles that were acidified in glutamate have a much bigger pH gradient, a much more not bigger, but more stable pH gradient, and they retain the pH gradient much longer even when you stop the proton pump. So there are several reasons for this. One would be glutamate is a better buffer for protons than chloride. So it holds on to the protons inside the vesicle, and that's why the protons can't get out. That is not the case. I won't show you all the experiments for that. But basically, the bottom line is, so this is how it works. This is why glutamate is really helpful for filling vesicles with monoamines and probably acetylcholine too, is that the proton pump makes the pH gradient. We inhibit the proton pump with baflamycin, and the protons will start to leak out of the vesicle. But the protons can't leak out of the vesicle unless an anion goes with them, or there'll be a negative charge left behind. So the only way for the protons to get out is for the chloride, the anion, to get out. And the bottom line is the chloride gets out pretty easily. So the protons get out easily because the chloride gets out easily, and vice versa. They go out together. But in the vesicles that were acidified with glutamate, what happens is it's a glutamate proton exchanger. The glutamate can't go out because it would mean pumping protons against their gradient. So it's just like a little ratchet to prevent the glutamate from going out. Because the glutamate can't go out, the protons can't go out. And it makes a more stable gradient. There are experiments to show this, but that's basically the idea. It's like a little ratchet that keeps the protons from leaking out of the vesicle. What I'm actually telling you is something that is completely counterintuitive, because this is the mechanism. So this is glutamate going into the vesicle, helping to make the vesicles acidic. Glutamate goes in, protons go out. The bottom line is that this mechanism works better to make the inside of the vesicles acidic than a mechanism where the protons don't go out. In other words, protons going out through this mechanism make the vesicles more acidic. Okay, this is the way it works. Okay, it's because of the equilibrium. And so it's just a little machine that does it. It locks it inside, and it can't go backwards very easily, and this is why it works this way.
So Robert, in vivo, or
Yeah.
in
Yeah.
a cultured neuron,
Yes.
how stable are the pH gradients?
You know, we look at them. We look at this kind of stuff when we do imaging of synaptic vesicle release, and they're relatively stable. This is what I'm showing.
People do those like you guys do
Yeah.
those experiments. You put in BAF,
Yeah,
and you're assuming that the vesicles
they're
that are
pretty
already there
stable
are
inside. But there's lots of other things going on there. It takes a while for the baflamycin to get in. So most of the time when we do those experiments, we're just blocking the protein that gets delivered to the cell surface, and we're hoping it doesn't leak in, actually. So it doesn't, most of the time,
I see.
technical stuff. So this is, if you add baflamycin to the outside of cells, it will block the proton pump of the vesicles that have undergone excitatosis. And then when they go back in, they cannot reacidify again. And that is instantaneous. As soon as they undergo excitatosis, the drug gets in, and they're kaput. Okay? And it takes longer for it to leak in and alkalinize the vesicles. It will eventually do it after 15, 20 minutes, but we do the experiments before that.
So Rob, what's PK of glutamate?
PK of glutamate is, I don't know, it's like 4 or something. And chloride is like minus 13 or something, but it's, anyway.
So it takes 2 hydrogen ions in as well.
This takes, it goes in as an anion. It does not go in its protonated form.
How does that work?
That's a good question. You know, there's some small amount of it around there. Yeah, sure, of course. It's a good question. I mean, but that's how the transporter, it goes in. Yes, 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 yes.
That's pretty interesting.
Yeah, I mean, it is how it works. So the bottom line of this mechanism is, you know, this is for the quantitatively oriented. If the mechanism by which chloride or glutamate gets into the vesicle, the anion gets into the vesicle, is a channel, you make a pH gradient that's 3 minus the log of the anion gradient. If it's this kind of mechanism, 2 chloride to 1 proton, there's some proteins that do that, it's 4.5. If it's a V-glut, let's say 1 glutamate for 1 proton, 1 glutamate going in, 1 proton going out, now your pH gradient is 6 minus this. So having more protons relative to the anion gives you a massive pH gradient. Okay, and it's because of the equilibrium. This sort of shows it. You know, shows that there's actually some math behind it. But yeah, anyway, it's a bit strange. But the bottom line is glutamate is helping to fill the vesicle with the monoamine. Okay. Okay. So now, so when we originally submitted that paper, there was a reviewer who said, well, you have a behavior when you knocked out VGLUT2 and dopamine neurons, but I don't believe there's VGLUT2 in dopamine neurons. I don't know how we've gotten a phenotype if there weren't, but they said, oh, but we don't see very much there. So we did experiments. Now the VGLUTs, all of them, go to the terminals of neurons. They don't go to the cell body. You can't label cells. If you see people claiming that this is a VGLUT1 or VGLUT2 positive cell because you see cell body labeling, I'd be kind of suspicious in almost all cases. It's almost always goes to the terminal. So you can't do it. You have to do in situ hybridization, or in this case, we got VGLUT2 GFP back transgenic mice. I'm pretty sure we got those from GeneSat. And so that's what we see here is in green in the midbrain of VGLUT2 GFP, now labeling the cell bodies of the glutamate neurons. And we stained for tyrosine hydroxylase in red, and this is a blow up, and you can clearly see that there are some TH positive cells which express VGLUT2. And we think, I don't know, there's a few percent at this particular age. I can't remember what the age was for this particular experiment, but subsequently another group has reported that it's 100% of the cells. I don't know if I'd go that high, but it's probably a lot of the dopamine neurons express some VGLUT2 even in adulthood, not just early in development. The other thing we noticed was something pretty weird, which was the midbrain's not supposed to have any glutamate neurons, but here there are a lot of glutamate neurons right here in the interpeduncular nucleus and in the medial part of the VTA, and I'll talk about those a little bit later. So, so there are other cells in the midbrain which are not catecholamine cells but are glutamatergic neurons.
So if you look for VGLUT3, I guess that's
It's not really. We thought that was originally expressed by the dopamine neurons, but it doesn't seem to be there. Serotonin neurons, and I should, because I'll probably forget, I should add that everything I'm saying here about dopamine neurons probably applies to norepinephrine, serotonin neurons, cholinergic neurons. So the RAF-A, for example, if you stain for VGLUT3, VGLUT3 is an odd member of the family. It's expressed by neurons not usually thought of as glutamatergic. Of course, these are not usually thought of as glutamatergic either, but VGLUT3, many of them are not thought of as glutamatergic. These include cholinergic interneurons in the striatum. And in the RAF-A, the densest staining for VGLUT3 by in situ in the brain is the RAF-A. It lights up like gangbusters. But it's not entirely clear that it's the serotonin neurons in the RAF-A.
I think the IPN as well is another one of those weird sites. It's got VGLUT3. So it's
Oh, it does? VGLUT3?
Yeah.
IPN.
And they project to the right, where you get the rest of VGLUT3, which breaks the striatum, where you get the cholinergic.
Well, we did some other experiments, which I wasn't going to show you, where we did some injections of channelrhodopsin to map the projections of the VGLUT2 neurons, and they had some other projection. But yeah, so all kinds of things. Yeah. So, but before I get to those other cells, these are experiments we did with the conditional knockout, and looked with Steve Rayport at the slice through the ventral midbrain and the striatum, and we basically saw that the EPSPs EPSCs he was recording in the medium spiny neurons would go way down, not completely disappear, in the VGLUT2 conditional knockout in the dopamine neurons, suggesting that, yeah, they're releasing glutamate onto those striatal neurons. But with Antonella Banchi, we did a much better experiment, which was to inject a conditional allele of channelrhodopsin into the DAC-Cree mice, and then we just cut striatal slices, and Garrett did in his lab, and here we shone light on the striatal slices. We don't have to worry about whether we're transecting them anymore. We're just looking at the terminals, and we can see nice, much bigger glutamate responses in medium spiny neurons. When we put on glutamate receptor antagonists, the responses go away. When we put on a combination of D1 and D2 antagonists, there's no effect, meaning that it's not dopamine being released by these cells, which is then causing other cells to release glutamate. It's direct release of glutamate from the dopamine neurons. Does not require dopamine release. Okay.
Did you guys look for asymmetric synapses
This is what this is the ongoing stuff.
So this is being released from the presynaptic terminals. It's not being released from the dendrite, because I remember.
No. There's dendritic release in the midbrain. Exactly what the source of that dopamine is, I'm not entirely clear. But no, this is axonal release, clearly, yeah. Because it's in a striatal slice. And I should say, I mean, we thought that one of the exceptions, I probably was going to forget this, one of the exceptions to this was the substantia nigra, because the substantia nigra doesn't express the VGLUT2, it's only the VTA. But then, of course, Bernardo Sabatini had this very nice paper suggesting that substantia nigra neurons co-release GABA as opposed to glutamate. So they all do it in some way or other. Okay. And then, consistent with that as well, this is looking in the ventral striatum. In the VGLUT2 knockouts, this is by voltammetry. The knockouts abolish this is the glutamate response, not the voltammetry. That's the next thing. This is the glutamate response to light. And then when we get rid of VGLUT2, it's gone. That's in the ventral striatum. And then in the dorsal striatum, we did the same thing. We did not see light-evoked glutamate release or response in the dorsal striatum at all. The wild type, what the controls are, the conditional knockout. But the problem is, of course, we had injected the channel rhodopsin into the VTA, not into the substantia nigra. But there's some spread of the virus, and we thought, well, so we also looked by voltammetry, and even though there's no glutamate response in the dorsal striatum, there's a lot of dopamine released measured by electrochemistry. So our channel rhodopsin was certainly getting into the projection of the dorsal striatum, and we're getting dopamine release, but no glutamate. So specific for the ventral striatum. Okay, any questions about that? So people have already asked the question, what's known about the release sites? So there's been, again, a long literature about dopamine neurons forming two types of synapses. The usual one was the en passant synapses, these boutons not connected with any clear postsynaptic element in the striatum, and then you get volume transmission. And then other people had reported with track tracing that there was more precise asymmetric synapses onto medium spiny neurons, typical glutamatergic synapses. And, are these, what's going on? Now, clearly our experiments say that there's some glutamate being co-stored with dopamine, but what I also just showed you is that glutamate can act as an independent signal for the postsynaptic cell. So it helps to promote vesicle filling with dopamine, and it also acts as an independent signal. So is there any evidence for release from distinct sites? Some of them are on the same vesicles, but is there any evidence of segregation of these release events? So this is quadruple staining of dopamine neurons in culture and hippocampal neurons in culture expressing either VMAT to identify the monoamine synaptic vesicles, and VGLUT1 in this case, which was what we were using back then, to label the glutamate vesicles. And the bottom line is when we compile a lot of this data, there's some yellow where they're co-expressed, and then there's some where they're only green or only red. And when we quantify all this, we expressed VMAT and VGLUT independently tagged with two different epitopes in hippocampal neurons. In axons, they basically 100% co-localize with a co-localization index here. But in midbrain dopamine neurons, there's clearly some glutamate release sites alone and some dopamine release sites alone. So there is some evidence of segregation. And what we would really like to know, and that's the next set of experiments that we're setting up to do, is in vivo, this is all in culture, in vivo, is there any evidence for distinct glutamate and dopamine release sites? And that's one of the things that we're working on right now. And that's not going to be so easy to do because the dopamine neurons express VGLUT2. And we heard about the thalamic projection to the striatum. The thalamic projection to the striatum is VGLUT2, tons of VGLUT2 in the striatum. So how can we distinguish the VGLUT2 from the thalamus from the tiny amounts of VGLUT2 which are going to be on the dopamine neurons? So there are various strategies to try to do that, and that's what we're working on now. One of the things we're working on.
If that were true, you might expect that different stimulus intensities would selectively...
Okay, so we already talked about that a little bit yesterday, but there is evidence in starburst amacrine cells in the retina and habenula cells. So the habenula system, where you have co-release of glutamate and acetylcholine, in that case, there's different responses. It was thought that one was released at a typical synapse, the glutamate released at a typical fast transmitting synapse, and other was more volume transmission. The acetylcholine was more volume transmission. But in the retina, there was pretty clear evidence that in that case it's acetylcholine and GABA, that they have differential dependence on calcium. And that would say that it's not just that the receptors are different distances from the same release site, but that the release sites are distinct. So I think that's what we're likely to find, but we'll see. And then the question is, why are the dopamine neurons releasing these two transmitters from different sites? What are the signals that they're really transmitting? Is it developmental? Is it for plasticity? Is the glutamate there to help plasticity for learning? It's not really clear.
So how do you think you could segregate those vesicles?
Well, we just want to see them by even just like my.
How could you segregate them into different areas?
Oh, how could you? Well, if they have different proteins on them, they can be targeted differently. Absolutely.
So, is there recognition proteins?
I mean, we don't know.
transporters?
Okay, so it's generally been thought that the synaptic vesicles expressed by a particular neuron, you see a whole cluster of them in the terminals, that they're all the same. But people have known for a long time that there are different pools of vesicles with very different properties. And I will show you a little bit of the data to suggest that VMAT targets to a different population of vesicles than VGLUTs. So actually have some functional data for them. So the mechanism, I do not know. So this is exactly the data. So this is an experiment. This is the kind of experiments we do a lot in the lab. This is VGLUT1 or VMAT2. And attached to them we have this what's called the ecliptic fluorine, which is a form of GFP which is shifted in its pH sensitivity. So at the low pH of synaptic vesicles, you can't see the fluorescence. It's quenched. With exocytosis and exposure to the higher pH outside, it becomes fluorescent. And in these experiments that we did is we added the drug baphlomycin I mentioned before. It acts from the outside, gets into the vesicle, and prevents the reacidification of the vesicle. So once we've stimulated release of these vesicles, so basically what's happening here is we stimulate 10 hertz for 60 seconds, and we look at the fluorescence. This is exocytosis. Endocytosis is actually occurring in this experiment, but it is invisible because we have blocked the proton pump, which reacidifies the vesicle and would quench the fluorescence. If I didn't have baphlomycin here, the fluorescence would go up, we stop stimulating, and it would go back down. Because I have baphlomycin, the vesicle can't reacidify. Those vesicles are trapped in their alkaline state and they're permanently fluorescent. So this is VGLUT1. This is a typical synaptic vesicle protein. We stimulate and it comes out, about half of it comes out with stimulation, and the rest of it is very difficult to recruit, even if we stimulate more. It just doesn't come out. This is what's called the recycling pool, and this is the resting pool. They have different names, but you just can't get a lot of these guys to come out. And this is VMAT. We think that many of these will actually be the same release sites, but different vesicles. In this case, VMAT, we stimulate the same amount, and only a small fraction of it comes out. These are hippocampal neurons expressing these. These are midbrain dopamine neurons expressing these. The VMAT just seems to go to a population of vesicles which doesn't like to come out that much. And this is all overexpression and culture and that sort of thing. But the prediction is that in vivo, it's going to influence how they respond to different types of stimuli. So that is actually the functional experiment. So I've told you two things. One is glutamate helps to fill the vesicle with the monoamine, but also there's some evidence that they can be released from distinct vesicles also. Okay. So, yeah, as I've said, promotes storage. Glutamate can act as an independent signal and maybe be released from different, actually, boutons or different vesicles. And then the last thing I'm going to talk about is this population of glutamate neurons in the midbrain, which is not catecholamine, which people didn't know about before. And the question is, in experiments that people did where they thought they were recording from dopamine neurons, maybe they weren't. Maybe they were recording from glutamate neurons. So we wanted to see, what do they look like? Are they different from dopamine neurons? Are they the same as dopamine neurons? So this is the work also of Tom Nasco. And what we wanted to do first is we wanted to just characterize the electrophysiological properties of these cells. So these are animals where we've labeled them both live with green in the glutamate neurons, red in the dacry mice driving TD tomato, the flock-stop tomato we heard about before. And so green is glutamate, red is dopamine. And then one of the characteristics of dopamine neurons are the IH, the hyperpolarization activated current. So here what we do is we hyperpolarize. These are medial glutamate neurons in the VTA. These are medial dopamine neurons in the VTA. And these are lateral dopamine neurons in the VTA. And this is the classic response. You hyperpolarize and you get this inward current down here with more hyperpolarization. And both the medial glutamate and the medial dopamine neurons have IH, this IH current, but much less of it. And they're in fact indistinguishable from each other. They have about the same. So whether they're glutamate or dopamine actually is no different here. What's different is the lateral dopamine neurons are different from the medial. Okay, so that's one thing.
Is there any difference between anterior versus posterior?
We did not do a systematic study of all sorts. There'll be lots of other things you could look at. And we did look at other parameters. We didn't look at other locations, but we looked at, D2 responses. So normally D2 agonists are going to hyperpolarize dopamine neurons. And here's a typical response of the lateral dopamine neurons to the D2 agonist quinpirole. You can see the hyperpolarization. When we do this with the medial dopamine neurons and the medial glutamate neurons, if you look at the compiled responses here, there's not a clear response. So only the lateral dopamine neurons seem to be D2 responsive. The medial dopamine are not very, and the medial glutamate are not very either. Yes?
What's the spontaneous firing rate of those medial?
We did not really. I would have loved to do that, but we didn't patch on them long enough before breaking in to be able to really know. So I would have loved to know.
Have you looked at other D2-like receptors? It's really interesting.
Yeah.
There's no D2 response to this. It's amazing.
Yeah, they're not particularly, and the medial dopamine neurons are not particularly either.
What about D4? Have you looked at other?
Well, this is, I don't know, actually, other people probably know better than I, quinpirole. It should hit them all, right? I mean, okay. The other thing that Tom did was he wanted to see, as Paul talked about before, where do these cells go, the glutamate neurons? So in this case, what we did is, this is what I showed you before. Green is glutamate, red is TH. In this case, we got VGlut2 Cre mice from Ole Keen in Sweden. So Cre is only going to be expressed in VGlut2 cells. Now, half the brain is VGlut2. So we have to be very careful with our stereotactic injections, otherwise we're going to get a lot of other cells. So we thread a lot of animals to get the few that I'm going to show you here. But Tom was able to get a few injections which were very precise in this region, in the medial VTA, which is what we were aiming for. And this is what he saw for channel rhodopsin. So channel rhodopsin fused to cherry. And this is the projection from what looked like these medial glutamate neurons, not interpeduncular nucleus, the medial VTA glutamate neurons. And I don't know, I didn't know what to make of this when he first showed me. This is the striatum, actually. And you'll see, but when we double stained it for tyrosine hydroxylase, this is what we found. And so actually the glutamate projections went to topographically distinct parts of the basal ganglia from the dopamine projection. They like fill in the interstices. They're not just mixed in. And we saw projections to the prefrontal cortex. We saw projections to the nucleus accumbens, which is what I just showed you. And so here is TH is in green. The VGlut2 cherry is somewhat distinct. You would expect some overlap because remember there's some VGlut2 in the dopamine neuron, so there's some that are yellow, and then there's some where there is actually more red alone, and consistent with that is also projections to the amygdala. So multiple projection sites, many places. Okay, yeah, prefrontal cortex. You can see that there's some that's clearly just glutamate, and others that are just TH, and there's a little bit of overlap. Okay, and this is another picture getting a better view of the ventral pallidum. There was a strong projection from these glutamate neurons to the ventral pallidum as opposed to the other parts of the, some other parts of particularly the dorsal striatum. Okay, so, okay. And yeah, this is the amygdala and also to the habenula. So you can see projections to the habenula as well as the amygdala. So the other thing we did with these injections of channel rhodopsin is we said, well, what kind of synapses are they forming? We expect them to be glutamate because we put these channel rhodopsin into VGlut2 Cre cells, so they should be glutamatergic because they should express VGlut2. And this is what we saw in the nucleus accumbens and also the ventral pallidum. We saw nice glutamatergic responses, in this case the depolarization blocked by the AMPA receptor antagonist, DNQX, ventral pallidum, something similar. And in some cases, we also saw GABA responses. We did not emphasize this before, but we also saw GABA responses. In some cases, they seem to be like the nucleus accumbens. They seem to be indirect. They had a longer latency, as if we were activating the GABA neuron, which was then releasing GABA onto the postsynaptic cells. But in other places, like the ventral pallidum, we actually saw in some cases, I'm not showing you here, fast GABA responses, suggesting these cells might release GABA as well as glutamate, or at least some cells release GABA as well as glutamate. But they're defined by VGLUT2, so we still don't really understand exactly what's going on with that. But you know, basically VTA glutamate neurons in the midbrain, they express less IH and less D2 responsive than more lateral dopamine neurons, but they're like medial VTA dopamine neurons. They project to these places, and they form, in general, excitatory glutamatergic synapses. What they do, we do not know. I mean, in terms of behavior, what does this whole population do? It's actually, this is a substantial number of these cells. So that's actually...
Just a shot of cocaine or electric foot shocking them and seeing if they...
yeah, well, this is one of the things that Tom in my lab convinced me while he was in my lab that would be too difficult. And, you know, but of course that's what he's doing now. That he has his own lab at UC San Diego. So, the problem is for these cells, so if we use the VGLUT2 conditional knockouts and we inject a virus into the midbrain, if we hit anything along the pathway, people will say, you get this cell, you hit that cell. So when we thought if we have a 10% really precise targeting accuracy and have to throw out 90% of the animals that we studied behavior, it would be difficult. But I'm sure there's a way, you know, he's going to figure out a way to do it. And he's mostly focusing on that. We're mostly focusing on the issue of glutamate versus dopamine release and whether they're released from similar different sites and what their role might be in development or plasticity. But that's actually what he's focusing on right now. So,
Robert, getting back to some of the questions that
Yeah.
we were listening to.
yeah.
Number 4 is your strategy as a new investment.
Yeah.
That's a really good
Yeah. I don't. Now that he's gone from my lab, I guess it's Opus. But anyway, yeah. So we did
No,
look.
that's his strategy.
Yeah, it was his strategy, not my strategy. So one of the questions was even going back to the conditional knockout of VGLUT2 in the dopamine neurons themselves. So not these purely glutamate neurons, but the dopamine neurons. You know, we had reviewers said, can you tell us what is the glutamate doing as an independent signal versus its role is filling the vesicle with dopamine? Oh man, I could not figure out how to do this. I mean, because if we block the filling, we block both of them. We don't know how to separate them. It was a nasty question, but anyway, it was okay. They let that go. But we did do experiments looking at those mice to see with operant conditioning. There are differences, even in the conditional knockout of VGLUT2, just in the dopamine neurons, there are some subtle different, I don't know how subtle they are. Tom probably didn't tell me how exciting they were. And there are differences with those animals as well.
So is there a compound that's like glutamate that will substitute for glutamate but then not have a response?
Yeah. Aspartate is not recognized. That would be a good one. It's not recognized by the transporter, so it wouldn't be released. It's a good idea. I don't know. Well, if it puts glutamate in the vesicle, it's going to do both things. But I still think there are experiments to do. We could look at animals with equivalent levels of dopamine production and see what they cannot do and compare them to these. But I don't know. Yeah, that was a difficult problem. So I actually, in the bigger perspective, I think it's the purely glutamate neurons. That will be perhaps the most exciting thing because that's all they do is glutamate. And we probably can activate it pretty specifically. So, anyway, yeah, so the people who did this were Tom did the conditional knockouts and the work on the medial VTA glutamate neurons. Xiaolin and Bibiana did some stuff on the trafficking of VMAT and VGLUT in neuronal culture and how they behave differently. And thanks for your attention.
I have questions, plenty, but it's kind of like off topic in
Sure.
a way, but you know the answer better than anybody else maybe. When I read about monoamines and vesicles, and I look at like preparations from the medulla.
Yeah.
They're referred to as dense core vesicles.
Yeah.
Now I think of dense core vesicles as peptidergic.
Yeah.
And so how do you differentiate those two? Are these dense
These
core
are synaptic vesicles. They're not dense core vesicles. It is always an issue because dense core vesicles clearly respond to different stimuli from synaptic vesicles. They generally require a lot more stimulation, and they can store catecholamines and acetylcholine, so they definitely can do that. But these vesicles that have been seen in, the axons of dopamine neurons, there are occasional dense core vesicles, but the dopamine is mostly released from smaller vesicles. And one technical thing is people used to stain with potassium permanganate, which used to make little dense cores inside the synaptic vesicles. But they're not dense core vesicles. They're really just synaptic vesicles.
So what they were studying from medulla,
those might have been dense core vesicles. I cannot comment on those. The adrenal gland, releases norepinephrine from dense core vesicles, and some cells clearly do. But this is synaptic vesicles. They're little vesicles. I don't think they have peptides.
And have you ever seen evidence of, say, VGLUT2 on a peptidergic vesicle?
It would be hard because there are not that many of them. We have not, we see not much evidence of that. Because when we do imaging, if it's on a dense core vesicle, they tend to give you a big response. You get a flash of light, a flash of fluorescence. Whereas the synaptic vesicles are so small that you don't see individual vesicles, you see the cumulative. And when we've looked at VGLUT2 fluorine, we don't see big things. So I don't know. We really haven't seen it much.
Do you think there's a way of coupling, say, input from prefrontal cortex and thalamus to the rate of uptake of dopamine? It's kind of cool that glutamate could control in that regard. So it might be a nice coincidence detector if you've got thalamic signaling or prefrontal signaling.
So the actual glutamate that would be released.
Yeah.
The glutamate would have to be taken up by the dopamine neurons. I don't know if they express any of the glutamate, plasma membrane glutamate transporters. They would have to do that. But there might be other ways to do it. I mean, it's certainly potentially a mechanism for plasticity. It's been shown that in some systems, I can't remember exactly now, but that glutamate that is released from excitatory input gets taken up. So they're glutamate transporters on GABA neurons. They take up the glutamate released by the excitatory input, convert it to GABA, and then you get more GABA released from the inhibitory neuron. So it's like some sort of homeostatic mechanism where glutamate gets converted to GABA after it's released to compensate, along the lines of what you're suggesting. But I don't know of any example for monoamines or anything. But it's possible. I mean, of course, I probably should have said it at the beginning, but one of the reasons why we were really interested in glutamate released by dopamine neurons is that it would be typical synaptic release as opposed to volume transmission, and that there would be this big difference and that they would serve some different role. And I still think we don't know the answer to that. We don't know if it does.
So in the DAT knockouts, do you get a big increase in the glutamate in the vesicles?
The DAT knockouts.
Because it's less dopamine.
Yeah. Well, I would say what we haven't done, which would be good experiments to do, would be to block the uptake of the dopamine into the vesicles, which you can easily do with a drug, reserpine, and see if it affects the glutamate responses. We never did that. We have to do that's an obvious experiment, because then maybe it'll shift. It'll become even a more potent glutamatergic synapse. I mean, it's not been done. It's the thing we didn't. It's one of the experiments we did not do with Antonello. Trying to get the paper out.
Is there regulation of the dopamine pool size in these vesicles?
Well, people studied tyrosine hydroxylase up the wazoo for decades as a site of regulation of catecholamine production. So that could be one. The vesicle transporter. In packaging. I mean, it's just not known. It's just
because a lot of, I think what you were talking about was, thermodynamically how much
Right.
concentration one can
Right.
get if you basically drive the system to an equilibrium.
Yes, right.
But if glutamate
Mhm.
is impacting this
Mhm.
filling process, you might imagine that there could be a lot of adjustment for the...
We did not look at these animals. It would have been better to do it in vivo. We looked at striatal slices from the conditional knockout and from the wild type, from the controls, and basically there was less dopamine released in response to stimuli in the striatal slices from the conditional knockout. But a better experiment to address what you're saying, we did not do it, would be to do it in vivo, because you can stimulate a lot more. And then we could see whether they have a bigger defect in dopamine release, particularly with prolonged stimulation. We never did that. That would be a good experiment because we could stimulate a lot more.
Because there seems to be a lot of homeostasis in the system if you go back to the DAT data that you showed, where you can deplete dopamine like crazy,
Mm-hmm.
actually, but you know, the release
Yeah.
didn't look that bad, really, in terms of
In the DAT, that's right.
Yeah,
Well, the release is pretty terrible. It looks okay there, probably because they stimulated the heck out of it.
yeah. Okay.
But it really should be reduced by 20-fold.
Okay.
Yeah, it's a massive, in the striatum. But it's hard to see. I mean, one of the problems for monoamines is there's no ionotropic response by the postsynaptic cell. The only place this has been, I don't know, is John coming this year, John Williams? No. Anyway, John has shown that the only place where you can get an iPSC with dopamine is in the midbrain itself. Dopamine neurons releasing onto themselves because they have the GERK channels which give you the fast response. And the striatum, the medium spiny neurons, don't have those. So it's even possible that dopamine is not acting in volume transmission, even in the striatum. But you can't tell now because the only receptors that you can monitor are not, rapid, ionotropic, they don't signal rapidly to channels. So the idea has been maybe even dopamine itself is actually acting more precisely. But because of the way we measure it, it's hard to tell.
So
So
could
people,
you make a mouse? Could you stick the GERK channel down there and hope that behavior isn't screwed up?
Chris Ford in Case Western Reserve, he's going to try doing stuff like that. Yeah.
It's just fascinating. So if you had to guess, what are the different factors that are regulating dopamine release in BTA,
Yeah.
let's say, and apportioning those factors either at baseline or dynamically
Yeah.
after reward anticipation.
Right.
What would you say are all of those factors, and how much of it is this role of glutamate to facilitate?
Well, we don't see, I think the dendritic,
all the different things.
Yeah.
Presynaptic
Yeah.
receptors and
well, the dendritic release, there's very little VGLUT2 in the cell bodies. You can't stain it. So I don't think the vesicles that are releasing dopamine from the dopamine cell bodies contain VGLUT2. That would have to be in the terminals. I frankly, although I've loved John's work over the years, I've never been entirely convinced that the dopamine release that he has observed in the midbrain aren't recurrent collaterals from axons. I'm never entirely convinced that isn't actually what it is, and it's not actually coming from the dendrites themselves. But anyway.
So at least there in midbrain, this isn't.
Yeah, I don't think the glutamate's affecting what's going on in the cell body.
terminal.
It's the terminals, yeah, pretty sure. Yeah.
This is Dave Goldman and Angus Nairn, by the way.
Okay, all right.
Okay, well,
Thank you very
Rob's
much.
going to be around
For a little while, yeah.
barbecues, so we go back. The barbecue should be down by the swimming pool. Think about the